Hi everyone, and welcome to our second video on DNA and RNA and nucleic acids. So today we will be focusing on protein synthesis, which will involve diving into RNA and the two processes involving RNA, which are translation and transcription. So first of all, we should learn about protein synthesis. So protein synthesis is important for all cells, and it would result in the production of proteins that determine all the characteristics of any living organism. So the instructions for protein synthesis are coded in the DNA, but RNA is the one that actually carries out the protein synthesis. So there are two processes involving RNA: transcription and translation. We see that here and here. Now, the information needed for these processes are coded, right? And they are coded in the form of triplets of the bases that we learned already. Right? We know the four bases, right? So they are coded in triplets, and the triplets have certain features that we will discuss very soon. But first of all, let's take a look at this table here. Now, the triplets actually code for amino acids, and there are about 20 amino acids, right? You don't need to know any of them. You don't need to learn them, right? But you can see all the amino acids there, right? I'm just outlining some for you. And very interesting, you could see that for most of them, they are coded by more than one triplet. You could see the triplets of bases, right? So UUG, UUA, UUC, that's triplets, right? And those are the nitrogenous bases that we talked about, right? So that is one of the properties of the bases, that they are degenerate or redundant, meaning more than one triplet will code for the same amino acid, which is actually very important when we look into diseases and mutations in our next class. It means that even if something were to occur in a base, it has a chance that it may not even affect the final product of the protein, because if, let's say, UUU, which coded for this one, and this last you change to a C, you might say, wow, well, that's going to code for a totally different amino acid. No, it doesn't. It actually codes for the same one, right? So that actually helps it out a little bit. All right, the other properties that all start with AUG, methionine. All right, all of the amino acids, all of the proteins, polypeptide chains start with AUG. That's also called the start codon. All right. Another one to note is that it is non-overlapping. So let's say you have a polypeptide chain that reads AUG, GUC, GAU. You have to read it in triplets, like how I just called it out. You can't say AU, GGU, UCG, GAU. That's not how it's done. It's non-overlapping. So 3 by one time, right? 3 at a time. And the last one is that it's universal. These triplets are present in every single living organism ever on the earth. Okay? So this is a nice little table that you could always refer to so you don't get confused. Okay? Right, so let's take a look at RNA now. So RNA is actually quite similar to DNA, but there is a few differences. Number one, the sugar is actually the ribose. Okay? It has a base of uracil instead of tyrosine, right? So you know how DNA would have the T would be tyrosine. In RNA, there's no T. Instead of the T, there's a U for uracil. Okay, that's the main difference there with the nitrogenous bases. RNA is only a single strand, so there's no double helix forming there. And there are 3 different forms which are all very important: mRNA, rRNA, and tRNA. So you'll see this when we go into translation and transcription. So messenger RNA is involved in transcription, while the tRNA, transfer RNA, is involved in translation, and rRNA is kind of in between, right? So let's take a look at the first main process, which is transcription. So this is the conversion of nitrogenous bases on a DNA strand into a complementary RNA strand. Right, so it's very similar to DNA replication, and there are 3 stages: initiation, elongation, and termination. I'll show you a picture of how that's done very soon. So let's just go through the 3 main stages. So in initiation, the enzyme RNA polymerase. We know about this one from the last class, right? So it binds the specific nucleotide sequence in the DNA, and this is called the promoter region. It makes sense, promoter, to start, right? Now, after the enzyme would bind, the two strands of DNA unwind, just like in replication, and these will act as templates for the RNA molecule. Remember, the RNA molecule is just one strand, right? So any strand would work. So elongation now, that was initiation, where the DNA unzipped, basically, right? So in elongation now, the RNA polymerase enzyme moves along the DNA molecule in the 5' to 3' direction. Same thing like replication, right? And then complementary base pairing will occur in the same fashion, except that uracil will be there instead of thymine. Okay? Now, termination now is where you finish, right? So at the end of the gene, there's a sequence called the terminator region. So you see how we have the promoter region, now we have the terminator region. So when RNA polymerase arrives at that point, it knows to detach and stop. Alright, and then the DNA will reform, form back its helix, and then you're finished. Okay? So let's see how it looks like here. So I have three main stages: initiation, elongation, termination. So in initiation, the DNA has already is just one strand now, right? It's already unzipped, and you can see that the base pairings are going to line up soon, right? So the U, instead of T, remember, U will bind to A. Remember, A goes to T and C goes to G. So instead of T for RNA, you would have U, right? And then in elongation, you have your complementary base pairing occurring: A to U, C to G, C to G, G to C, those kind of things, right? And then finally, termination will occur. The mRNA chain would have formed, and the DNA would wind back up into its helical form. Okay, very easy. Right, so that was transcription, and we have a little table here outlining the differences between transcription and DNA replication, so you can just take a look at that. It's very self-explanatory. Right, so the last thing today is translation. So this now is how the codons and amino acids are actually formed, and this uses the transfer RNA, tRNA. Okay, and you'll see how it's done. So translation is the conversion of a sequence of bases on mRNA into a sequence of amino acids on the polypeptide chain. So we're actually going to make amino acids now. So there are quite a few things that you would expect that are needed for this, right? So you have mRNA, ribosomes, RNA molecules, the enzyme peptidyl transferase, amino acid tRNA synthase, as well as amino acids and obviously energy, right? And you'll see how all of these work together in a diagram soon enough. Right? Now the function of the peptidyl transferase, it catalyzes the peptide bonds between the amino acids. Remember, amino acids will form a polypeptide chain via the peptide bonds. We learned this in earlier on in proteins, right? So that's what the peptidyl transferase does. The amino acid tRNA synthetase, it catalyzes the chemical bonding of a specific amino acid to a specific tRNA. So it the tRNA is the transfer molecule, and amino acid needs to bind to the tRNA, right? So that enzyme helps that to happen, right? So the tRNA is like a vehicle. Okay, so I have some definitions here so you don't get confused. So the triplet code refers to a sequence of 3 nitrogenous bases. The anticodon is simply the sequence of 3 bases on a tRNA, and I'll show you how that is done. It's actually just the opposite of what is on the mRNA. Okay, just like how DNA and RNA would have complementary base pairing, it's simply complementary base pairing on the between RNA molecules. And finally, a codon is simply is a sequence of 3 bases on mRNA. Okay, translation also has the same 3 steps: initiation, elongation, termination. So very easy to remember. Okay, so initiation now. So this is the beginning. So the tRNA molecule, and the tRNA molecule always has the start codon AUG or methionine, right? So it associates with a small ribosome, and that forms an initiation complex. Big term, but it just means we're going to start there. That's what the initiation complex means. okay? And the initiation complex is going to move with the RNA molecule now in a 5' direction, and the large ribosomal unit will now be added on to complete the structure of the ribosome. Right, so just so you don't get confused, I have the picture here. So you can see the large ribosomal unit there, the 70S ribosome, and you can see that our mRNA begins with AUG. So obviously our tRNA now is going to bind to that AUG. But this is what I mean by the complementary base pairing, right? So because you have AUG, the tRNA molecule now is going to do the same thing that the DNA and the RNA did. So A would be U would be A, G would be C. So this is what you would see on the tRNA, right? So I hope that makes sense. So you could continue practicing and going along. So if this was GUA here, that T would be so GUA. So on the tRNA, you would get C, then you would get A, and then you would get U on that tRNA molecule, right? So the tRNA molecule is simply going to, and next one is going to form here like this. You draw it, so it looks like a T, right? It's very easy actually to remember. So another T will form, and it's going to add another amino acid there to the methionine that's already there. Right, so it's going to add one there. As you can see in step 3, it added a valine. Now when that happens, this one will simply go away. It's going to pick up another, it's going to move essentially to this spot, and then it's going to pick up another codon, do the same complementary base pairing, get this amino acid, and bind it to the valine. And that's how it's going to make an entire polypeptide chain. Alright, that's just a summary. So let's go back to the second step now of elongation. So the peptide bond, as I said, is going to form between the first two amino acids. So we had methionine, and then the second one is valine. And again, the reaction is catalyzed by the peptidyl transferase enzyme. Okay. Now the first amino acid, tRNA molecule, the tRNA molecule will break the bond between the tRNA and the amino acid. And then, as I said, the first tRNA molecule will simply be free to leave that site and pick up an next amino acid. Alright? And the ribosome, the big pink thing, is going to move codon by codon, triplet by triplet. It's going to move and start generating the polypeptide chain. And finally, termination. So how does this whole setup know to stop? Well, there are many stop codons, right? So let's go back to the table. All the way here. So you see all these bold ones here? So there are lots of stop codons. So anytime you meet the codon UAA, UAG, or UGA, that will signal everything to stop. That means the polypeptide chain has either finished or some bad mutation has caused a stop codon to occur. And then in that case, that polypeptide chain would be incomplete. But that is a story for another video. So in this video, we kind of looked at the two main processes in RNA and protein synthesis. I hope you guys enjoyed the video.